Good evening, Pittsburgh and universe. This is JC on a Bike. My name is JC, and this is Journal Club on a Bike. Journal Club on a Bike is a production led by GigaOhm News. And GigaOhm News is news brought to you by a biologist. It's high resistance, low noise news brought to you by a biologist. And Journal Club on a Bike is where this all kind of started for me, trying to spread the word about exciting neuroscience papers. And then in the beginning of February, a blog titled Harvard to the Big House came to my attention and started me down a long, deep rabbit hole of gain-of-function viral research and SARS-CoV-2, the modern-day coronavirus pandemic. The thing that about this blog that struck me was that I just hadn't computed the possibility. It had never really dawned on me that the pandemic that was brewing in China, that we had already seen footage of the massive sort of draconian lockdown that occurred in China in January, where they were bulldozing piles of dirt in front of tunnels and welding the exit doors of certain big buildings shut so that people could only come into their residences through one door. And we saw all these videos of people getting kidnapped on the street, it looked like, by authorities, putting them in boxes and taking them away. And so in January, we were already watching crazy stuff happen in China, and the WHO was already giving us information about what was going on in China, and most of that information was, of course, as we know now, quite wrong. There was never any real serious talk about airborne transmission, human-to-human transmission, or what could be done about it until the end of February. In January and February, nobody was talking about a pandemic. Nobody was talking about a very dangerous virus. And the only people who really knew that anything interesting was going on were the people that were living in Wuhan, because they were seeing it firsthand. And so we were forced to kind of reconcile our government and our authorities and the WHO telling us that it was no big deal, and yet social media and the things that we were seeing were showing us a very different picture of a communist government that looked like it was in panic. And so the things that were going on in parallel at this time when I read Dan's blog inspired me really very quickly to start diving deep into the literature and become the sort of investigative biologist that I have evolved into as a result of this coronavirus pandemic. And I'm happy to say that that's what happened, right? I was just a mild-mannered neuroscientist working on my own little problems in science, and I was happy to be doing that. But the biologist in me, the deep-seated biologist in me, realized that Dan was really onto something, and with even a cursory search, it started to become very obvious to me that I had missed something, that I had forgotten about what happened in 2011 and 2012 because I had heard about it before. And what led me to connect all these dots in the early weeks of February was just searching and finding articles by Lynn Klotz, who has been working tirelessly at trying to raise awareness of gain-of-function research and raise awareness of the dangers that gain-of-function research represents to humanity. And he's not the only one writing these papers, but he is one of the foremost sort of authorities on the statistical computations that describe the likelihood of a laboratory release, and they are not zero. None of these calculations result in a zero. And what only became really obvious later in the exploration was the sheer amount of speculation with regard to what happened here had its roots in many incidences that have happened in the past. In addition to the 2009 flu pandemic, which was discussed as a possible laboratory leak, the SARS original release also discussed as a laboratory leak. The 1977 influenza pandemic is known to be a laboratory leak. And so this was all getting me really fired up. I mean, and also trying to understand what they learned about SARS and MERS in those months and years after 2003 when SARS broke out, and then 2012 when MERS was discovered. They have these long-standing publication records that have investigated and charted the investigation into these early beta coronaviruses like SARS and MERS. And what I got very disturbed by was that a lot of the things that they were hyping as crazy and scary and new and, you know, this novel coronavirus is nothing like we've ever found before. And I kept finding papers from the original SARS and from studies looking at the MERS viruses that we knew all this stuff already. We knew about the cytokine storm and what role it plays in severe influenza and SARS. We knew already about the cytokine storm. We knew already that coronaviruses can be neurotropic. We knew that SARS was neurotropic. We also knew that a lot of the viruses a lot of the coronaviruses, when used, portions of them are used, or when antibodies are artificially developed against the spike protein of SARS, it was already shown that the spike protein is dangerous for that, and oftentimes antibodies generated to a portion of the spike protein will cause antibody-dependent enhancement. And they saw that already with vaccine candidates with the original SARS. So none of these problems, none of these symptoms, none of these what are painted as very novel aspects of SARS-CoV-2 are actually not novel at all. And so you have to actually question, why in the world would they go on TV and tell me that this is scary and novel when it's actually not so scary and novel? And so then March comes along, and in the early part of March, the awfully written, totally conflicted, not at all scientific proximal origin of SARS-CoV-2 comes out in Nature. And so immediately the floodgates are opened, and everybody is quoting this paper and citing this paper as the authoritative evidence that this is not a laboratory-oriented or a laboratory-originated virus. And this paper has been destroyed by a number of authors separately, showing that the claims that they make are wrong, that even the citations that he makes in that paper are sometimes incorrect in terms of what he says the citations show you. And so the disingenuousness of this manuscript cannot be underestimated, and yet it is still cited today by many articles as the primary authoritative evidence that this was not made in a laboratory, did not come from a laboratory, couldn't have been leaked from a laboratory. And about a week later, a guy by the name of Michael Osterholm goes on Joe Rogan's podcast and starts to explain how we are not smart enough to have come up with this virus. We are not smart enough to have come up with the sequence, and no gene jockey is clever enough to have thought of making this virus. And these words struck me as extremely chosen, extremely well crafted, not just pulled out of the air, but actually saying something very carefully. No one could have designed this virus. No gene jockey would have ever chosen this sequence. That statement is not the same thing as saying this is not a laboratory virus. Not at all. And on that day, when I saw that video and I listened to him speak, I was screaming and yelling like I'd only done one time before in my life. And that was when Building 7 fell. And after I saw the podcast with Joe Rogan and Michael Osterholm, I was ranting and raving and screaming and yelling in my backyard, just like I had done after 9/11. And Building 7 fell due to fire. So this really got me fired up. I really wanted to understand how big of a lie this really was. And so the first thing that Michael Osterholm reminded me of, and Dan really reminded me of, is the 2012 papers in Nature and Science that showed cultured animal passage of avian flu to enrich it to be airborne in ferrets. This was done in 2 labs, one in the Netherlands and one in America, in 2012, and it sparked a huge debate about gain-of-function research simply because these were viruses with certain pandemic potential. And they were created in a laboratory where they didn't need it. We didn't need it. There was no reason to make an airborne H5N1, but they did it anyway. And this sparked this debate again, reignited this debate about gain of function. And as you peel back the layers, it's just extraordinary. To say that we couldn't have made this virus, it's extraordinary. And that's where the primary literature really nails it. Because in the year 2000 or 2001, we made an infectious clone of the polio virus from scratch. In 2002, Ralph Barrick's own lab used similar techniques to generate a cDNA clone that could generate infectious RNA of a coronavirus. A mouse coronavirus, but it was a beta. It's a group 2 coronavirus. Mouse hepatitis virus. They made it from scratch. They proved that they could do it in 2002. And then in 2003, interestingly, the same lab decides to resurrect from a sequence a lost and extinct original SARS virus clone to prove that these techniques could be used to generate infectious RNA from a sequence. So that's just one technique, set of technique papers from 2002 to 2004. It's now 2020, folks. How much happens in 15 years? Well, I'll tell you, a bunch, right? Because there's papers after that come with even further beautiful sort of fine-tuning of the techniques necessary for seamless ligation and making viruses without deletions, without substitutions, exactly the sequence you want. And of course, there's all kinds of well-published coronavirus backbones, coronavirus recombinatory systems that allow the ease of swapping parts of the viral genome between viruses so that you can investigate what it is they do. And in principle, this is just biology. except you're not working with cultured brain cells in a slice, or cultured brain cells in a dish, or you're not working with a slice of mouse brain that has absolutely zero possibility of becoming a pandemic, of killing millions of people. But if you're playing with viruses and mixing and matching parts of viral genomes, then what we know about virus is simply not sufficient to know and be able to predict what sorts of gains of function might happen when combining certain pieces of certain viruses with other viruses. Who knows what happens if you mix certain support proteins or enzymatic proteins for replication with different parts of another virus, and together an emergent function could result that we just can't predict because we just don't know enough about viruses. And of course, we haven't even got to 2015 and the famous cloning paper where they are taking a SARS backbone and adding the spike protein from a bat virus. This is the paper that everybody zeroed in on as being the one. But all this paper really shows you is what they can do. And once you know what they can do and how they do it, then you only need your imagination to know that they could have made SARS-CoV-2. The 2015 paper was cited by me as shady because a viewer of mine from New Zealand had looked at the online version and noticed that the earlier online version of the paper existed. It wasn't the Nature medicine form. And that paper, that manuscript, had 6 or 5 for sure references that weren't in the Nature online version. And she and I found this interesting because the papers that they leave out are not insignificant. Usually when Nature says, you can only have 35 references for this paper, then you would cut the ones that, contribute the least to the understanding of the paper. And these 5 papers that were omitted for one reason or another on the online version on Nature's website are all papers that give pretty big clues as to how much they know and how much they've figured out about coronavirus infectivity. 2 or 3 papers zero in on the receptor binding domain and which amino acids are important for ACE2 binding in humans versus other animals. One of those papers looks at the ORF, the open reading frame 6 protein, which is involved in transferring the mRNA from the cytoplasm to the nucleus, and then this regulation of this process can affect viral transcription. And so they analyze this to show that if they put a certain open reading frame 6 protein into another virus, that you could increase its infectivity. That paper, and then another one from a textbook, details the use of human airway cultures that are a mixed culture of cells with the idea of simulating a human lung in a dish. And this ORF, the open reading frame 6 paper, uses that culture preparation. But also another reference in this paper is to a textbook chapter about human airway cultures that is actually written by a guy who works in the same university as Barrick. So without a doubt, they were using human airway cultures to enrich this virus. And human airway cultures are human airway cultures. You want to argue about what they can do with those. With human airway cultures, you can select for a virus like SARS-CoV-2 from amongst a bunch of viruses that might not be very good, easily. You cannot underestimate how powerful that technique might be, especially because of how little we actually talk about it. So those papers are omitted on purpose, I'm sure. It's a choice so as to not call attention to those crucial points in their methodology that would give any bench scientist a very good idea of the breadth and depth of their capabilities. But it wasn't really until the clue in the Nature paper from Sheng-Shi Li on the 2nd or 3rd of February, until we figured that clue out. And that clue was that the RATG13 consensus sequence was sequenced from a virus that was collected, a sample that was collected in the Moyang province of Yunnan. So this led us down a rabbit hole that the seeker pretty much nailed shut when he found the master's thesis that described the 6, not 3, the 6 miners which were infected with an unknown pneumonia in 2012 in this very cave. And so in 2012, Sheng-Shi Li's lab and the Army and the CDC of China, they all went there several times over the next 2 years, and sampled the bats, sampled their feces, and made lots of papers, including a couple papers that described the miners, which we found. But when we found the master's thesis, then it was over. Because the master's thesis describes the medical condition of these miners, and there isn't a doctor on planet Earth that wouldn't call what they had COVID-19. Except it was COVID-12, right? And so the postulate is that there's no good explanation for why RATG13 wasn't published when it was found. It was one of 2 bat coronaviruses that was a beta coronavirus found in that cave, where they were looking for beta coronaviruses related to SARS. And they found one. And then they left the sample in the freezer for 6 years, they say. And then later, they changed their story over this Chinese-led virology seminar last weekend. They changed the explanation to it was sequenced in 2018. And then that sequencing reaction finally used up the sample. So that's it. And so now here we are with Nikolai Petrovsky's paper showing with computer modeling that the binding energy of SARS-CoV-2 to the ACE2 receptor is just absurd. And it doesn't look like zoonosis at all. And Alina Chan shows the same thing with maybe a bit better analysis, because she also looks at the changes that occurred in SARS in 2003 and demonstrates the mechanism of zoonosis by showing you that SARS was not exquisitely adapted for humans when it jumped, and then it slowly mutated. In the first few months it was in people, and those mutations were, a lot of amino acids changing. And as you're aware, we're only talking about single amino acids changing in SARS-CoV-2, and right away making a big hype out of them. And so the only way that biologically makes sense is this virus had evolved in some other system or some other place before being released onto the population of Wuhan. There's just no other explanation for it because the virus isn't changing. And this is extremely damning evidence. It's not damning evidence that maybe PBS NewsHour can explain to you, but it's definitely damning evidence. It's evidence that doesn't support the zoonosis. And to finish it up on the bike, just to let you know, in the first one, and this is explained very well by Alina Chan's papers, the last figure, is that in the first viral outbreak in 2003 of SARS, we were able to deduce the immediate host of the virus very, very early. We found the civet analog very, very quickly, and the agreement in the sequence was like 99%. So we were pretty sure that virus was the precursor to the SARS that jumped to humans. And it took them several more years to find out that maybe the precursor to the civet virus was in bats. And now, curiously enough, we're in the opposite scenario. We think, according to the WHO and China and the NIH, who funded the research, we think that we know where the virus ultimately came from, but we haven't found the intermediate host that explains the virulence yet. And my argument is the most parsimonious explanation for that is that host is in a laboratory. Hey, thanks for joining me on this ride. This was a swing for the fence. I was trying to hit a home run here. It's been a long time since February. We've gone through a lot of iterations on the hypothesis, on the theory, on the sort of biological explanation for what's going on with the coronavirus. And I think we're at a stage now where people that are in charge are hearing our cries and they are hearing our claims, and they are either having to purposefully ignore us or having to address them to a certain extent so as to sort of stop the snowball from rolling down the hill. But I think we've already started it, and I think the snowball's already rolling, and there's not much to worry about anymore other than to keep harping on it and keep spreading the biological truth to the masses. So that's what this bike ride is about. It's about trying to give you an introduction into the history of laboratory gain-of-function research, the kinds of things that have been talked about and discussed in the past, and the kinds of things that have been done in the past which sort of belie the circumstantial evidence, underpin the circumstantial evidence, which suggests that there's more than enough methodology from already a decade ago to describe a way that SARS-CoV-2 could have resulted from laboratory work and laboratory accident. The only question is now, how are we going to legitimately investigate this so that we can really understand where this virus came from? All that being said, I don't think it's possible that they have created something that is outside of the design parameters that Mother Nature has set forth for coronaviruses. What that means is that this coronavirus, albeit novel, is moving through our population in exactly the same fashion that all coronaviruses move through our population. And we are seeing these very typical curves from epidemiology where the infection starts, it goes very quickly, and then at some moment there is a plateau because there aren't enough people that are naive to the virus around the people who are infectious to continue the exponential climb. At some moment, the asymptomatic portion of the infection takes over. And we know this from all other coronaviruses. We know it from influenza. Asymptomatic infection by viruses is not new for coronavirus, regardless of what they tell you. And this is another thing that they are combining all of these myths and falsehoods into a package to try and make you believe that SARS-CoV-2 is really this brand new, never before seen nightmare from nature, and it's really not. And I think as we move forward, if you keep your eye on Sweden, and you keep your eye on New York, and you keep your eye on the real numbers in Japan and in all these other countries, especially the countries that are using hydrochloroquine, you're going to see a very normal respiratory virus outbreak, given the fact that it's novel, given the fact that it's quite infectious and nobody has a real strong immunity to it. This is what we would have expected. This is what Knut Witkowski said. This is what Wolfgang Wodak said. And so these people have been right, and we need to acknowledge that they have been right. We have gone through the pain, but we've probably gone through more pain than we should have gone through, simply because locking down oftentimes spreads out and extends what would otherwise be a very quick flare-up and a flash-out. So this was a bike ride. My bike rides have been few and far between lately, so I hope you enjoyed it. If you liked it, like and subscribe. Please share this with your friends so I can get more than 2,000 subscribers, for goodness sakes. And again, thanks very much for watching. This is JC on a Bike. Gig Ohm News is high resistance, low noise news, brought to you by a sweaty biologist on a bike. See ya. What in the nine hells are you doing over here, you little chicken shitters?